In the second part of this session, Dr. Katie Janeway, she's a colleague and a medical oncologist in the Pediatric Oncology Group here and at Boston Children's Hospital. She's going to continue the discussion on GIST and focus on wild-type GIST. She has both presence in the lab as well as presence in the clinic, and I'm honored to give her the stage.
Thank you. Well, thanks for having me speak with you today. I'm going to speak about advances in wild-type and pediatric GIST. And you'll notice that pediatric GIST really just refers to an age group, right? So pediatric we typically think of as meaning 18 years and younger. And wild-type GIST has typically been used as a term that's been used to describe the absence of certain features that are common in GIST, which are those mutations in KIT or PDGF receptor alpha, which are the sort of hallmark biology changes that we see in GIST. And we're trying actually to get away from these terms. So they're very imprecise. So it's sort of imprecise to describe GIST just based on the age of a patient or based on the absence of something that we see in the more typical form of GIST. And so actually I'm going to mostly focus on describing what we now call SDH deficient GIST. And at the end of the talk, I'll get back to describing how that relates to pediatric and wild type, which are terms that have been used quite commonly until now. So I'm going to talk about what it is, how we diagnose it, what causes it, how it's treated. As I said, I'll get back to sort of telling you how it relates to the pediatric and wild type GIST, and then I'll tell you a little bit about where we're going from here. So what is SDH deficient GIST? To answer that question, first I just want to review a few features of typical GIST. So typical GIST has in the tumor a change in a gene called KIT, PDGF receptor alpha, or BRAF. And that change is typically what we think of as sort of causing or initiating the GIST and making it multiply. In this tumor, the cells tend to be spindle-shaped. Spindle just means that it's stretched out and long. And also there's a protein, which is called SDHB, that's present in the tumor cells. SDHB, I'll get to this a little bit more in depth later, is involved in cell metabolism. And the way that we demonstrate that the tumor has this SDHB is by something called immunohistochemistry. Immunohistochemistry is something that's done in the pathology lab with a tumor. And I'm just showing you a picture of it. So this is GIST, typical GIST, how it looks under the microscope. And this is how it looks when it's had SDHB immunohistochemistry. It's brown. And brown is basically showing you that the SDHB is present or there. So what is SDH deficient GIST? So that protein called SDHB is not present in the tumor cells. And we can demonstrate that, or the pathologist can demonstrate that by immunohistochemistry. And so I'm showing you again the typical GIST up here where you see the picture and then you see the brown staining. And in these SDH deficient GIST, when you try to get this brown staining to show up, it actually doesn't show up. In these tumors, the cells tend to be epithelioid or more round shaped, and you can actually get a little hint of that by looking at these three pictures here. This is the typical GIST with the spindle cells, and these are the rounder epithelioid shaped cells. And in these tumors, we don't find KIT, PDGF receptor alpha, or BRAF mutations. So who gets SDH deficient GIST? So the reason that I became interested in this group of tumors, or I started studying these group of tumors, is because 85% of the GIST that occur in children are SDH deficient GIST. Some adults do get SDH deficient GIST. It's a very small proportion. We actually don't know the number yet, but we think it's about 5% or maybe less of adults with GIST will have SDH deficient GIST. And most, but not all, of whom develop SDH GIST are female. So it's much more common if you have SDH deficient GIST to be female as compared to male. The reverse is not true. So if you're female and you have GIST, you're not highly likely to have SDH deficient GIST. Again, if you're older, it's still very uncommon to have SDH deficient GIST, even if you're female. So how do we diagnose a GIST as being an SDH deficient GIST? Well, the first thing is that a pathologist sees the typical appearance of the tumor just on that plain slide. So they see that the cells are rounder shaped, And also the way that the tumor grows tends to be in nodules. So they're sort of these clumps of tumor cells that are all clumped together. Then the pathologist can do SDHB immunohistochemistry. And then finally, to sort of verify that we're really dealing with an SDH deficient GIST, the tumor is tested for KIT, PDGF receptor alpha, and BRAF mutations. So having identified this group of tumors, the next question was, what causes SDH deficient GIST? And it turns out that this SDH thing is actually composed of 4 different proteins, and one of those 3 succinate dehydrogenases, or SDH genes, has an error or mutation in all the cells of the body in a small proportion of people who have SDH deficient GIST. And these genes are called SDHB, SDHC, and SDHG. And as I said, this error or mutation in one of these SDH genes is present in about 15% of people who have SDH deficient GIST. The cause in the other 85% of people with SDH deficient GIST is still not known. So let me explain a little bit more by what I mean about there being an error or a mutation in one gene in all the cells of the body. Oops, go down one more. Yep. So because the error in the gene is not just in the tumor, it's in all the cells in the body, it means that it can be inherited or passed from one generation to the next. And when we're talking about SDH, that error or mutation, what that really causes is a tumor predisposition syndrome. And that means that because the gene abnormality is present in all the cells of the body, it has the effect of increasing the risk of developing a different particular kind of cancer. In the case of SDH, those cancers are GIST, very rarely, paraganglioma, pheochromocytoma, renal cell carcinoma, and medullary thyroid cancer. Everything else, even though there is this error in all the cells of the body, everything else about the person is normal. They look normal, they have a normal life, et cetera, et cetera. And actually, it turns out that with this particular abnormality, the risk of getting any one of these cancers is actually quite low. So if you look back in the family, even though the gene is inherited, most people in the family won't have had any tumors or cancer. So let's just tell you a little bit more about what SDH is. It's part of something called the electron transport chain, and it actually is what helps your body use oxygen in the process of converting food to energy. And when SDH is abnormal, the cells can't use oxygen properly, which promotes this cancer development. And I'm just listing here those tumors that I told you about that can occur in people who have this SDH mutation in all the cells of their body. So getting back to the terms pediatric and wild-type GIST, how are all these GIST types related? These are really subtypes of GIST. I'm leaving out now all of the typical GISTs, okay? So I'm just going to talk about pediatric and wild-type. So if you start with pediatric GIST as a big circle, 85% of pediatric GIST will be wild-type. We take wild type here. These are wild type GIST. Those are the GISTs that don't have the KIT, PDGF receptor alpha, or BRAF mutation. You see that there's some group of wild type GIST that are not pediatric, meaning these are wild type GIST that occur in adults, people over 18. See the next one. So then SDH deficient GIST, which is what I just told you about, basically every pediatric GIST that's wild type will be SDH deficient. So these two circles overlap. This middle circle here overlaps completely with the SDH. That's probably not true for adults who have wild-type GIST. Adults who have wild-type GIST, some of them will have SDH deficient GIST, and some of them will not. And then finally, 2 other terms I'm not going to talk more about, but you may have heard, Carney triad and Carney Stratakis dyad, are really just subtypes of SDH deficient GIST that can occur either in children or in adults. So how is wild type or GIST treated? Okay, so now I'm going to go back to using the term wild type because we don't have specific information about clinical behavior or treatment that's specific to SDH deficient GIST because we've only recently discovered that that entity exists, or we've recently defined it. And most of what we understand, we've understood from focusing on wild type or pediatric GIST. So this slide is thanks to a collaboration with the National Institutes of Health, and this is called the NIH Pediatric and Wild Type GIST Clinic. And basically what I'm showing you, so this is before we can really understand what we should do for treatment for pediatric and wild type GIST, we need to understand how the disease behaves. And we don't have that much information about it because it's a rarer form of GIST. And so what this slide is every person who's attended this NIH Pediatric and Wild Type GIST clinic, each person is shown as an individual bar, and the light blue is showing you the age at the time of diagnosis, and the dark blue is showing you the current age. So anything below the red line, with the light blue below the red line, which is this group, are people who had their GIST diagnosed by 18 or younger. And to the right of this are people who have wild-type GIST who were diagnosed when they were older. And the important take-home message from this is that, for example, if you look at this person, they were diagnosed at 18, but now they're in their 50s. Okay? And so what we think about this wild-type GIST is that although it does tend to come back and it does tend to spread to lymph nodes, maybe perhaps even more than the typical GIST, people can live for a long time with wild-type GIST, even if it's present in a metastatic form. So in terms of treatment with imatinib, sunitinib, and other kinds of tyrosine kinase inhibitors, studies show that imatinib and sunitinib do work in adults with wild-type GIST. Imatinib's probably not quite as good as for GIST with KIT and PDGF receptor alpha mutations. There's only about 45% of people with the wild-type GIST who take imatinib who will have tumor shrinkage. Sunitinib is probably not as effective in shrinking tumors, but seems to stop growth for a prolonged period of time. And I'm very happy to say that for the first time, we're actually going to have clinical trials focusing on this rarer subtype of GIST. So an international study, which is a phase 1-2 trial, as James just talked about, the different phases of trials, of sunitinib in pediatric and wild-type GIST. And basically what this trial is trying to figure out is what is the best dose of sunitinib to use in a younger patient with GIST. Believe it or not, we actually don't know that. In patients who are 18 and younger, we really don't know what dose of the drug to use. So the dose of sunitinib was studied in children, but it was studied in children who had other cancers and had previously received a lot of strong chemotherapy medication. And the dose that ended up being determined to be safe is probably too low for somebody with a GIST tumor. And so what we're doing in this study is we're looking at using the drug just in young people with GIST and trying to allow us to go to a higher dose in a very controlled sort of studied environment. We won't go to a higher dose than what is the equivalent dose for an adult, but the problem is that the pediatric study determined quite a low dose, and we think it's too low. The other thing that this study is doing is it's looking at the activity of sunitinib, that's what the phase 2 part comes from, in pediatric and older patients who have the wild-type GIST. So we're focusing this study on wild-type GIST. It's actually the first time that there's been a clinical study for this group of patients. So another interesting area to think about treatment for pediatric GIST is looking at the insulin-like growth factor 1 receptor. This is a receptor tyrosine kinase like KIT and PDGF receptor alpha, and pediatric and wild-type GIST have very high levels of IGF-1R on their cell surface, as do many other cancers. Mutations are not present. IGF-1R is involved in normal cell growth, and it's thought to stimulate cells to grow and divide. And this is another example. This is the second clinical trial that's really targeted at this subgroup of GIST patients. And this is a phase 2 trial of lincitinib, or OSI 906. This is targeted at adult patients, but the reason for wanting to study this drug, which is an IGF-1R inhibitor, in GIST is because of this subgroup of patients. And neither of these studies are quite open for enrolling patients yet. The sunitinib trial will probably be the first site to open it, and the lincitinib trial, I think, is it's starting to go through the regulatory process of being opened here. So in terms of what we're working on for the future, so the NIH Pediatric GIST Clinic, which has been really a great way to start to understand this rarer type of GIST, is continuing. And really, I'd like to echo James' thanks of people who take the effort to participate in research. We really have learned a lot of what we know about this group of GIST thanks to people traveling to Washington to participate in this clinic. So that's really been very important for us. The second thing we're working on is to better understand the causes and roles of SDH and GIST. And so there's several laboratories, including ours here, working on better understanding causes of the SDH deficient GIST, and that 85% of people who don't have that error or mutation in SDH in all the cells of their body. And then finally, we work pretty hard on educating providers, pediatric oncologists and medical oncologists about this rarer form of GIST, because you sort of need to understand it and think about it a little bit differently than typical GIST. And I'd like to thank the Cancer Research Fund for its efforts. Really, all of the advances we've been able to make in understanding this rare subgroup of GIST has been through funds like the Cancer Research Fund. So. Questions? Yes.
I'm sorry. Yes. Do we have any idea of how many actually facilities will be running the clinical trials for the pediatric?
For the pediatric sunitinib?
Yes.
So, yes. In the U.S., there are going to be, I don't know the exact number, but it's about 8 to 10 sites that will be running that trial. In this area, we'll be running that trial. Memorial Sloan Kettering will be running that trial. And then there's probably going to be one site in each region. So there's going to be a site in Florida, one in, hopefully one in Seattle, and Texas.
Okay, so we will also know who's running the clinical trials on that as well. And we don't know yet about the dose. I mean, we know for certain that the 50 milligrams of the sunitinib was totally very toxic, even for the adults. So the middle, which is 37.5, would probably be a startup dose that the clinical trial.
So for the young patients, we're required to start at the lower dose that was determined to be safe in those patients who got a lot of previous treatment with chemotherapy. So we're required to start at that dose because that's the only dose that we know to be safe. But what we're doing is if somebody is tolerating that dose well, and it's not working against the tumor, we're going to increase the dose within the setting of the study so that we can understand if that's the safe thing to do in young people. That's really the purpose of the study.
Right. Okay.
Is to really understand the dosing better. Because as you know, if you don't get the dose right, the drug doesn't work. Right. And I think probably the current dose that we know of is not the right dose for children with GIST.
Many adult patients that have been on sunitinib have also used the 37.5, but alternate with the 25 milligrams as well. So is that something that the clinical trials will probably also consider doing as well for the kids because they are totally a different, mechanism with their bodies. So
that's right.
Yeah.
Right. And we'll be checking, that trial allows for checking levels as well.
Yeah, okay. Thank you.
You're welcome. Any other questions?